I wanted to talk about the synthesis and application of DNA-protected silver nanoclusters. So as we know here, so in this place, so they present the missing link between metal atoms and the nanoparticles. So as we know here, so this is the cluster. So the electronic and optical properties of metal cluster depends greatly on their size, in particular in the nanometer range. So when the size of the metal cluster is further reduced to about 1 nanometer or less, down to a few atoms, the bond structure becomes discontinuous and is broken down into discrete energy levels, somewhat similar as the energy level of molecules. So we can see here we call the fluorescent noble metal nanocluster, also called the noble metal quantum dot. So when particle size of noble metal nanoparticles becomes comparable with the Fermi wave length of an electron, say for example half nanometer for gold and silver. So metal nanoclusters, we say, become the molecular species and the discrete state with strong fluorescence can be observed. So here data is the energy level gap. So it's equal to EF. This is the Fermi energy divided by the N. N is the atomic number in the particle. So if data equal to 0 here, that's a bulk matter. And if data smaller than KT, K is Boltzmann constant, T is absolute temperature. So that's the metallic cluster particle. So if data is is larger than KT, so this is the insulating cluster particle. And if it is much larger than KT, so this is atoms and molecules. So this is just to show you the electronic structure transition process from bulk matter to nanoclusters. So the characteristics of the fluorescent noble metal nanoclusters. So first, compared with quantum dots, the smaller size of the noble metal clusters are benefit for the biolabeling. Second, the fluorescence band can be turned from blue to infrared ranges. And good photostability and low environment toxicity and precise synthesis methods. So in theory, only noble clusters smaller than silver 40 or gold 55 behave fluorescence emission, and such small clusters are difficult to characterize by TEM. In this case, mass has been applied in the characterization of them. So modification, why we want to study. So fluorescence noble metal nanoclusters with the biomolecules such as DNA, peptide, or protein as scaffold or a class of novel inorganic-organic hybrid fluorescent materials and have great potential in the bioimaging and sensing applications. So this talk is concentrated on the synthesis and application of fluorescent gold and silver nanoclusters, mainly based on the results in our laboratory. So first, I wanted to talk how to prepare. So we have developed a technique for high-yield synthesis of DNA monomer protected silver nanoclusters and density functional theory predictions of their photoluminescence properties. So this is the procedure for the preparation of the silver nanocluster. So here, if we use the alcohol at 0 degree, we get the intermediate. We add DA, DT, that's the adenine, silane, and the DC cytosine, and the DG qualine. So in this case, get an intermediate, and after the addition and for NADH4, this is powder added. Then just after some time, say for example, several hours, we can get several nanoclusters. Another way is that in alcohol, so in this case, we can prepare to get several nanocluster with NTB. So N can be, for example, ATP, CTP, GTP, and TTP. And here we add the solution added. Finally, we can get several nanoclusters. So it's quite simple in this way. We would like to say the technique use the DNA monomer and also its triphosphate as the scaffold. Now we get the result. Here, this is the TEM imaging of the several nanocluster protector with different scaffold, such as this one is DA, D-adenine, DNA, AB, and CD, that's the DT. And the EF in the next here, the EF, so that's the DC, and the GH, DG, and their particle size distribution here. So we can see here, this is the size is about 2 nanometer. That's just in the range of the clusters. Now here show you the typical TEM imaging of several nanoclusters by DATB here and TTB here, and CD as the DCTB and the DGTB, and also by the atomic AFM, atomic force microscopic imaging of the several nanoclusters synthesized with different scaffold as DATB and DTTB and DCTB and GTTB, these pictures. We also studied with multi-top. So we can see here for the DA, DT, and the DC and the DG. So all this data, we can see that it's quite significant that AG9 is a big peak. Of course, we can get also the other number, for example, AG11, AG13, and so forth. But AG9 is significant, is the main signal. And also, we use this multi-top to study the DATB, DTTB, DCTB, and the DGTB. We get also, in this case, the AG9 is significant. And we also, by different spectroscopic methods, for example, here this is photoluminescence interference. So I wanted to stress that in this case, so if DA, DT, DG, so this is very small. But for DC, it's a significant peak, so excited and emitted. And here, this is absorption, UV visible. We get this, and also studied by infrared, Fourier infrared, to get such in different cases, and with several nanocluster to compare. So from all these results, we get the strong interaction between the DC and the CF9. So this is the ground states, and this is the excited states of the silver and the cytosine. And the optical ground state geometry of DCG AG9, and this is the optical excited states geometry of the DC AG9 complex. And here, this is the molecular orbitals we have calculated, and the electronic contribution of the relevant excitation state for the DC and AG9. Now we get this photoluminescence spectra, so that we have calculated by Gauss 09 software to get the modulation, get the result is 590, while experimental data is 591. So it's quite agreement. So further, we can study to calculate this value. So this is the reduced density gradient, and here this is the electron density, and this is the sign of the second Hessian eigenvalue value. That's data with delta 2. So by calculation, we get the value here is 0.025. As we know, if less than this value, it's a hydrogen bond. Less than this, just here, this is a van der Waals force, van der Waals bond. So it means that AG9 compound with the cytosine is quite stable between the hydrogen bond and van der Waals force here. This here just shows the interaction between silver and the oxygen and the nitrogen of the cytosine. So this is the first part. So we can get technique to prepare the silver nanocluster with mononucleotide. Then we want to switch to talk about the single nucleotide mutation identification. So Professor Dixon from Georgia Tech, they study used the single-strand DNA to prepare the silver nanocluster many years ago. So now we think why we cannot use the double strand for the preparation. So therefore, we developed the technique with the double strand to prepare as the scaffold to prepare the silver nanocluster. So here in this case, the strand A, this is the target segment of the home supporting hemoglobin beta chain, we call the HBB. Yes. And B, this is the mutation of this. So target segment of the variant of HBB. And strand C, we call the prober. So that's the designed cytosine loop, 6C loop. Yes, the inserted prober strand. So here, this is the sickle cell alanine mutation point. So if it is the T, so that's the sickle cell anemia. So we can see that if use this double strand, strand C and B, so we can get the photoluminescence quite clearly. However, if C with A A is the normal. In this case, no fluorescent. So this is just to show you that the form of two different cytosine loop is inserted DNA duplex working as the synthetic scaffold to generate fluorescent silver nanoclusters for the identification of the sickle cell annealing gene mutation. So in this way, just here change one gene from T to A, so we can get that with or without fluorescence. So this technique can be used for the identification of the sickle cell annealing gene mutation by the generation of fluorescent silver nanoclusters. So we get the result like this. This is excited, this is emitted. This is at the 520 nanometer. This is 572 nanometer. So it's quite clear that we can also get this the bright color for the fluorescence. And the control experiment and selectivity we have studied. So from here, I just want to stress that, so if strand C with strand B to form the duplex, so we can see that here, this is a quite significant peak for the absorption and also for the fluorescent, for the electrophorescence. So it means that only in this case, that's strand C with strand B to get this. So this is just to show the mismatch, differences between these three cases. Because strand C and D duplex and strand B here, just here. Only strand B with strand C to form very significant peak. In a similar way, we can use, for example, here, 6-base anthrax laser factor sequence. So here, this is a mutation point. So if this is A, so this is just in this series anthrax laser factor. And if this is indifferent, for example, ATCG or YATCG, this D is a duplex. This D is a duplex comprising proper strand. So we can get different cases, different results like this. For example, if X is A and Y is T, so we get this quite significant. However, if X is C, CG is still differentiate. This is significant and the difference with the other cases. And here, if X is T, Y is A, TA, so we get this significant remarkably. And in this case, if X is G, GC, YC, GC, this is also significant. So it can differentiate for the AT or TC. This is quite significant for the. So when X of strand D is complementary to Y of strand E, the corresponding DNA duplex stabilized silver nanocluster exhibit the highest photoluminescence emission. The highly sequence-dependent fluorescence silver nanocluster formation in DNA scaffold can identify different types of the single nucleotide mutation. So the background DNA duplex region adjunct the cytosine loop has impact on the silver nanocluster formation. And when the 4 completely complementary DNA duplex scaffold generated photoluminescence from silver nanocluster differed in both photoluminescence intensity and photoluminescence band. Conclusion. So this work expands the available capping DNA scaffold of fluorescent silver nanocluster from single-stranded oligonucleotide to hybridized DNA duplex. And second, the highly sequence-dependent formation of the fluorescent silver nanocluster in this duplex scaffold was able to identify specifically a typical single nucleotide mutation, the sickle cell anemia mutation. The identification of based on this strategy was further extended to more generally type single nuclear mismatch, such as anthrax laser factor sequence, etc. The third example I want to show you the photo-induced electron transfer of DNA silver nanocluster modulated by G-Quadruplex Hemin complex for the construction of versatile biosensor. So if we have the silver nanocluster with the G-quadrate sequence, so by the addition of Hemin, so it can be folded as a G-quadrate. So in this case, we found that this is quite strong, this is weaker. It means a quench effect. So this is due to the photo-electron transfer effect. So photo-electron transfer induced for quenching effect. So I wanted to say, so in this case, because the ferri and the ferro, and this is quadruplex, this is the redox as the electron acceptor. So therefore, the photo-electron transfer here to quench, not to the ground, but to this step, and therefore it's the quench effect, and it's related to the concentration, for example, of the Hemin. And here just show you that the photograph and the TEM. So this is the low light, this is the ultraviolet at 365. And by the TEM, we measure to get 1.14 nanometer. This is just in the range of the nanocluster. So UV visible, we have studied. So 2 peak, one is 400, second is 560. So this is just to show you the DNA silver nanocluster Hemin. And the purified DNA silver nanocluster here. This is, yes. So we can see here, this is the excitation and the emission. So it's in this case, so this is brown is quite small. However, if it is in this case, so excitation at 560, so we get remarkable. This is significant. And this is here excitation at this. This is not active. And C, so purified DNA, C is also not, C is here, yeah, is purified, and DE is the separated of the silver nanocluster particle. So therefore, this is just to show you the typical peak of the silver nanocluster and purification at excited potential excited wavelengths at 400 not just this at this wavelength is active. And here also show you the experiment that the excitation spectra, and here this is the emission spectra, and B is the DNA silver nanocluster. But if in the presence of the potassium, it's no influence. But in the presence of the potassium and the Hemin, so this is due to the photoinduced PET-induced electron transfer. So therefore, the quench effect is very significant. And compared with this, compared with without CFR synthesis now. Here, this is just show you in the different concentration of Hemin. So concentration Hemin from 0 to 2 micromole in this case. It can be used for analytical purpose. And this is show you the specificity. So if in the presence of the Hemin, the quench effect is quite clear, significant. However, if for the other things, say, for example, porphyrin, IX, so this is no such effect. So it shows that it's quite specific. And it is also selectivity. So, for example, this is the distance, length of the distance between DNA silver nanocluster and the G-Quadruplex plus Hemin complex. If 3 pair base, then 5 or 9, and so. So it means a part of this place, so it decreased quite a lot. And this shows you the CD curve. So for the potassium and for the lead. So this is parallel, this is anti-parallel. So potassium is here quite clear, and however for the lead is not so sensitive as in the presence of the potassium. And here should show you the lifetime to study that. So here, in this case, if the silver nanocluster, and this is silver Hemin in the presence of the Hemin, and here in the different concentration of the Hemin, we can see that. So if in the absence of the Hemin, it is 2.18, and in the presence of the Hemin, it's much faster. This is 768 nanoseconds and so forth, just to show you. And this we have compared the CV DNA silver nanocluster and the CV of the separated silver nanoparticle. So you can see that's the difference. And this, the cyclic voltammogram of the Hemin in phosphate buffer. So quite, you see the reversible is different, the peak position as the silver nanocluster. So we have calculated this. So E excited states, E ground minus Planck constant, velocity of light, and wavelength, and this is the charge quantity. So by this, finally, we can calculate this value is 1.45. So if it is going to the ground like this, but in this case, because of Hemin here, so therefore, this is the electron acceptor. So just come to here. So that's the reason why we can get the quench. Also, we can use this quench effect for the determination of the Hemin or some other similar properties. Now, the first application, we can use this for the DNA detection. For example, if we have this DNA, so this DNA, if the complement with this, so due to the Hemin, by the addition of Hemin, it's folded. And here, with this, can form the duplex, and then here, so to the photo-induced electron transfer, therefore the pinch effect here. So this can be used for the analysis of nucleic acid. In this case, this just show you experiment that, for example, free, and then in the presence of the Hemin, so decrease, and in the presence of the Hemin, and in the presence of the target DNA and the Hemin, even down to this. So just to show you the pinch effect can be used for. And this is quantitative analysis. So we can see that by the addition of DNA, it decreases, and this is a concentration relationship. And this is also show you the selectivity is very good, because, for example, it's T1, T2. T1 is target, T2 is the one mismatch DNA, T3 is two mismatch DNA, and this is four mismatch DNA. I think time is over. Yes, another application for the ATP detection, it's a similar way. However, in this case, we use the ATP aptamer. So in that case, here is the hairpin can broken to, in this case, and then also to have the photo-induced electron transfer. In a similar way, in this case, we can have the concentration relationship for the deter. And this show you the specificity. So in the presence of UTP, CTP, GTP, we can get. So it is very simple and not requiring separation and troublesome procedures. It avoids the complex labeling process with DNA silver nanocluster as the fluorescent probe and the G-Quadruplex Hemin complex as the acceptor. It is a multiple sensing platform only with a little change on the sensing system. The fourth project I don't want to talk, so acknowledgement, the financial support, and thank you for your attention.